So we're going to switch gears a little bit. We were talking about metabolites and things at a basic science level. We're going to keep on the basic science level, but now we're going to switch over into more a gene type level. And one of the things that we've tried to do at the CRF is to have a broad swath of research projects that we fund. And Dr. Porter is going to talk about some of these types of projects specifically that he's working on, but that the CRF supports. And he is a research professor in the Lick Lab in the Department of Pharmacology and Physiology at the University of Rochester in New York. And Dr. Porter's work focuses on investigating the use of anticodon edited tRNAs as a platform for therapeutic for nonsense associated diseases. And his current work is also focused on developing viral and non-viral approaches for active in vivo delivery of these gene therapeutic, gene modulating entities. And if my memory serves me right, actually, I'm not sure if you were at our last meeting in Rochester. I think you were. I know that Dr. Leck was as well. And it's kind of exciting because our last CRF meeting was actually in Rochester. These gentlemen came. We formed this nice collaboration and association. And so the presentation we are about to view is a direct result of last meeting. And I think that that's very, very exciting. So thank you very much.
All right. Yeah. Thanks for the kind introduction. And thank you to the organizers for inviting me to represent the Lewick Lab and talk about some of our work we've been doing. And first, I'd like to thank all the other speakers for kind of introducing Rep 1 and function and how choroideremia works. For my talk, I'm going to focus a little bit more on sort of the fundamental molecular biology associated with this, and so hopefully I'll do a good job of explaining the basics to everybody so that we can all follow along. And so for the purposes of this talk, I want to talk a little bit about Rep 1, the protein. So what is a protein? Protein is a polymer. A polymer is just a larger molecule that's composed of repeating units. So in the case of a protein, it's composed of amino acids. And so shown here on the left is the primary sequence of Rep 1. So it's basically just all of the amino acids written as the one-letter code for each of them. And one thing to keep in mind for proteins is that the primary sequence defines the chemical structure of the protein. And so the chemical structure is shown here on the right. And the chemical structure is important because that's sort of what defines the activity of the protein in the cell. So as I mentioned, proteins are composed of amino acids. And so what's shown here is Anyway, what's shown here are the amino acids, the chemical structure, the name, as well as the one-letter amino acid code for each of them. There's 20 amino acids that are composed proteins in cells, and amino acids are linked together by peptide bonds, and that's what this string of amino acids is what composes a protein. And so just zooming in a little bit on the Rep 1 structure, on the right here, you can actually see with these amino acids linked together, you can actually look and see some of the amino acids themselves. There's a glutamine, lysine, methionine, and arginine. And so some of the key takeaways from this section is that proteins are composed of amino acids, they're linked by peptide bonds, and that the protein primary sequence defines its chemical structure, and that sort of defines its function. And so the next big question is, how does a cell, quote unquote, know how to make a protein? And to answer this question, we turn to what's called the central dogma of molecular biology, and this basically just governs the flow of information in the cell. And so the information that's held in the sequence of DNA when it undergoes transcription is then the DNA is used as a template to make mRNA, and then during translation, the mRNA is used as a template to make a protein. And so if we look at an example of this, we have a string of 9 nucleotides that make up a sequence of DNA. And whenever it undergoes transcription, it then is transcribed as mRNA, and so the only difference there is that a T becomes a U, but it's basically the same sequence. And then if we look at the process of translation, we see that we have 9 nucleotides, and then that's translated into 3 amino acids. And the reason that it goes from 9 to 3 is that the way that translation works is that a triplet nucleotide is sort of the functional unit and is what's called a codon. And so that's why 9 nucleotides gives you 3 amino acids. Now, if we think a little bit more deeply about codons and what are all the possibilities for a codon, so there's 4 nucleotides that are used for mRNA. There's 3 positions in a codon, and so overall there's 64 possible combinations of nucleotides that could make up a codon. And that's sort of often represented as a codon table that's shown on the bottom right here. And the way that a codon table kind of works is that the first position in the codon is on the left side of the table. The second position is on the top, and the last position is on the right. So if we were to think of CGA, you can look at C on the left side, go over to G on the top, and then A on the right side, and you see that CGA encodes an arginine. So that's kind of what is also shown at the left there. Okay, so key takeaways for this section. DNA is a template for mRNA. mRNA is a template for protein. mRNA is divided into codons, these triplet nucleotides. And each codon specifies an amino acid. One thing I didn't mention yet is that there's 64 possibilities. 61 of them are codons that code for an amino acid. 3 of them are codons that code for a stop codon. And basically the stop just means to terminate protein synthesis. And so then the question is, okay, this is fine conceptually, but molecularly, what's actually happening in the cell during translation? And so one thing I kind of left out when I was describing translation originally is that there's some other factors involved in translation. One of those is the ribosome, which is sort of a macromolecular machine that is able to read the mRNA and then link amino acids together to form the protein. And there's also aminoacyl tRNAs, or ACE-tRNAs, that kind of serve as an adapter molecule in translation. And so if we take a closer look at this, we can see in the bottom right, there's a ribosome that's doing translation. So it's reading an mRNA that's shown there, and then kind of poking out to the right side, there's these little balls. That's the growing polypeptide chain. And so basically the mRNA is linking amino acids together and forming a protein. And if we zoom in kind of to the interior of the ribosome and take a look at what's happening, you can see up top in purple, there's an mRNA, and there's a leucine codon in this case. And then we can also see there's a tRNA, and at the top of the tRNA, there's what's called the anticodon. And so there's actually molecular complementarity between the codon and anticodon. So U pairs with A, U pairs with A pairs with U in this case. And on the other end of the tRNA, there's an amino acid, in this case a leucine. And so functionally what's happening here is the tRNA serves as the adapter because it goes from the leucine codon to the leucine amino acid. And so then the question is, what can go wrong here? And that's really what we're focused on. And so a change in the DNA sequence is called a mutation. And so in this particular case, we're showing a particular type of mutation, a single nucleotide variant. So this C in that middle codon is being changed to a T at the DNA level. And so when this is transcribed into mRNA, what happens is now that what was a CGA codon is now a UGA codon. Unfortunately, UGA codes for stop. And so rather than having aminoacyl tRNAs come in and continue translation, there's actually release factors that come in, and this actually dissociates the ribosome, the mRNA comes off, and the protein is released. And so because this is happening now in the middle of the protein, we call this a premature termination codon, in that it's happening too early. And so if we think about this in the context of CHM, we have a rep1 PTC variant. There's no stable functional rep1 produced. And so what that means is that the Rab proteins are not properly modified or trafficked in the cell, and ultimately this results in CHM. Okay, so the key takeaways here: mutations are changes in the DNA. PTCs result in a stop codon in the middle of rep1, and loss of rep1 causes CHM. And so, the next question here is, what are ACE-tRNAs, and how do they function to rescue proteins during translation. And so if we kind of go back to our original picture of translation, we have the leucine codon, the tRNA, and if we want to make an ACE-tRNA, and so it's kind of actually in the name there, ACE is anticodon edited, and so if we look at the anticodon, we've changed what was AAU to now ACU, and effectively what this does is it makes the anticodon complementary to the PTC codon, and so effectively what we're doing is rewiring translation, so now instead of UGA meaning stop, UGA would mean leucine in this case. And so if we think about this sort of at the whole codon level, we think about what codons could be one nucleotide away from a stop codon, there's actually 22 possible amino acid to stop variants. Now we start to think about this from an ACE-tRNA's perspective. There would be 10 families of amino acids that we would need to make an ACE-tRNA for. And one thing that we've sort of realized as we've been developing this technology is that advancing all of these, 10 different therapeutic modalities to the clinic at the same time probably wouldn't be the most effective. And so we sort of had to reframe our thinking for the broadest applicability, and that we maybe want to go to what we sort of call a platform approach. And so in this case, it would be basically one ACE-tRNA family that could be used for any PTC. And when thinking about this, we really, leucine kind of stuck out to us as a good potential for this because it's able to efficiently suppress PTCs. It does it for all 3 stop codons. And we've also been able to optimize the tRNA sequence so that it's actually a much more potent suppressor. And so effectively what we're doing here, just showing this codon table, is we're just rewiring all PTCs as a leucine. So one thing to keep in mind with this platform approach is that while it is codon specific, it's actually protein and amino acid agnostic. And so this can cover many PTC sites, and in theory it could cover multiple genetic diseases as well. And so one thing we want to look at was what's the actual applicability of this platform approach to CHM. And so we cloned 10 of the most common Rep1 PTCs, and then we looked at expression of full-length Rep1 when we transfected these into cells, either with or without the platform ACE-tRNA. And so we saw anywhere between 20, in most cases, anywhere between 20 and 100% rescue of these PTC variants. And we were very encouraged by this because we anticipate that somewhere in the 20 to 50% of rescue is where the therapeutic threshold would be for CHM. And so where are we going with this? Really now we're focused a lot on delivery because we still have to be able to effectively get the ACE-tRNAs into the tissue that is affected. And so we're currently employing PTC mice to look at this, and I would say the more mature approach that we're looking at right now is using AAVs that have kind of been discussed before, basically using AAVs to deliver the ACE-tRNA. But I think where we're really going in the future is non-viral approaches. And ACE-tRNAs are very interesting as non-viral cargoes because the whole gene itself is so small. It's on the order of 200 base pairs, where most genes are in the orders of several thousand base pairs. So we're kind of in a different regime in terms of therapeutic cargo. And so we're looking to minimize the size of these vectors. We're able to make them fully synthetic, and we're working on new targeted delivery of them. And so with that, I'd like to thank the sources of funding for this, particularly the Thurcell and Hillier Families Research Award and the CRF for funding this research on CHM, and also our other funding sources for funding for working on the underlying technology associated with this. I'd also like to thank members of the Lewick Lab, particularly John, Zach, and Genesee. Zach and Genesee are two talented undergrads in the lab, and they've done a lot of work with this. Zach cloned and tested a lot of the Rep1 variants, and Genesee worked on non-viral approaches for rescuing CHM. And this is them in the middle there presenting their work at the U of R Undergraduate Research Symposium. So with that, I'd like to thank everyone for your attention, and I'll be happy to take questions at the end. Sorry, I'll repeat the question. So we're talking about nonsense mutations. What other types of mutations are there? So there's missense mutations, and that would be you have a change in the DNA that you go from one amino acid to another amino acid. That's a possibility. You can have splicing mutations where I didn't really get into it, but there can be other issues with making the mRNA. So this particular approach would not be amenable for other mutations beyond nonsense mutations.